Welcome to Peptide Proof. We follow the science, the evidence, and the market behind peptide innovation, from research and synthesis to therapeutics, applications, and industry trends. Today, we're looking at Palmitoyl Tripeptide-38.
When we talk about peptides in skincare, the conversation usually stops at the big three: relaxing muscles, signaling collagen, or remodeling damaged tissue. But this raises a really important question the next time you're analyzing an ingredient list. Is your peptide actually rebuilding your skin's full architecture, or is it just fixing a single wire? Today, we're exploring a major paradigm shift, moving away from targeted single-action peptides to a new standard of broad-spectrum intervention.
To really understand this shift, think of standard linear peptides, kind of like an electrician who arrives to fix a single wire. One input gives you one specific output, like collagen I. Palmitoyl Tripeptide-38, on the other hand, acts a lot more like a general contractor. It actually triggers a coordinated, simultaneous rebuild of 6 distinct extracellular matrix components. It doesn't just show up to fix the wiring, it brings the whole construction crew.
Here's our roadmap for today's explainer. We'll cover the Matrikine mechanism, the 6-protein output, the clinical and in vitro proof, a highly necessary formulation reality check, and finally, strategic synergies.
Section 1. The Matrikine mechanism, engineering the signal. Let's look at exactly how the biological blueprint of this molecule actually works.
To build a solid scientific foundation here, we first need to define what a matrikine is. Simply put, a matrikine is a naturally occurring peptide fragment that gets released when your body breaks down extracellular matrix proteins. And these aren't just waste products, they are highly specific signals. When tissue degrades during normal cellular turnover, these fragments basically tell your fibroblast cells that the matrix is being dismantled and it's time to rebuild. It's essentially the body's own internal maintenance feedback loop.
So let's break down how this specific molecule is architected for cosmetic application. The Matrikine signal itself is a precise lysine-valine-lysine amino acid sequence. This directly mimics a natural breakdown fragment of type 1 collagen. But here's the catch. On its own, this tripeptide is way too water-soluble and too large to actually penetrate the skin. That's where the delivery vehicle comes in. By attaching a 16-carbon palmitoyl fatty acid chain, this lipid tail transforms the fragment into a lipophilic molecule. That allows it to successfully bypass the stratum corneum's lipid barrier and actually reach the living fibroblasts beneath.
This structural engineering enables a really precise 4-phase playbook for matrix reconstruction. First, lipid matrix diffusion allows the 650 Dalton molecule to partition right into the skin. Second, the sequence successfully docks with those targeted fibroblast receptors. Third, instead of just kicking off a single linear pathway, this docking activates a multi-pathway program. And finally, this broad activation commands the architectural assembly of new scaffolding, hydration, and anchoring proteins.
Section 2. The 6-protein output. Broad-spectrum results. Let's dive into the actual structural proteins that get synthesized when this biological signal is delivered.
This breaks down that broad-spectrum advantage we just talked about. Palmitoyl Tripeptide-38 hits 6 specific targets. For structural integrity, it synthesizes collagen I for tensile strength, collagen III for elasticity, and collagen IV for the basement membrane. Then, for organization and adhesion, it produces fibronectin to scaffold the collagen, hyaluronic acid for deep hydration, and laminin 5 to anchor the epidermis directly to the dermis. So you're getting a complete, comprehensive structural scaffold, rather than just forcing one single output.
Section 3. Clinical and in vitro proof. Validating the mechanism. Now we move from the theoretical mechanisms into the concrete data. And I want to start with a standout metric for you to consider: a massive 124% increase in hyaluronic acid synthesis.
That metric comes directly from 48-hour in vitro research, and it really demonstrates the sheer catalytic power of this peptide. Alongside that 124% jump in HA, there's also a roughly 80% increase in collagen type 1 synthesis compared to untreated controls. The strategic insight to take away here is crucial, because Palmitoyl Tripeptide-38 works at absolute microdoses, specifically a 3 parts per million threshold. Even a relatively low 5 to 15% penetration efficiency through the skin barrier delivers a sufficient active payload to trigger this massive systemic response.
When we look at the published clinical data, the results completely back up this fast-acting kinetic profile. The gold standard study from Lintner and colleagues in 2020 tested just a 5 parts per million concentration over 56 days. Using quantitative 3D optical imaging, they recorded an 8 to 9% reduction in roughness, alongside measurable drops in redness and improved tone homogeneity.
Studies by Sanz in 2016 and Herndon in 2015 also confirmed statistically significant improvements in wrinkles and overall skin density. Now, while these studies tested the peptide in combination formulas, the rapid 1 to 4 week onset of visible results uniquely aligns with those in vitro matricine signaling kinetics we just observed, rather than your standard slow collagen building.
Section 4. The Formulation Reality Check. It's time to examine exactly why so many retail products fail to deliver on these clinical promises.
It usually comes down to concentration math. Let's look at what we call the concentration funnel. A supplier typically sells this asset as a solution containing about 100 parts per million of the active peptide. A brand might formulate their product using just 1% of that supplier solution simply so they can highlight the peptide on their marketing label. But the mathematical reality? 1% of a 100 parts per million solution yields exactly 1 part per million of active peptide in the final formula. That falls fatally below the 3 to 5 parts per million threshold established in the clinical literature. For it to actually work, the product needs to use 3 to 5% of the supplier solution.
Beyond the concentration math, environmental stability is paramount. This peptide operates in a physiological golden zone, right between a pH of 4.5 and 6.5. That's where it maintains its optimal structural integrity. If you expose it to a highly acidic environment, say anything with a pH below 3.5, the amide bonds in the peptide backbone actually start hydrolyzing. The palmitoyl tail detaches, and the active acid degrades entirely before it can ever penetrate the dermis.
So, to protect the structural integrity of your peptides at home, you need to follow a few clear rules of separation. If you're using exfoliating acids like AHAs or BHAs, apply them first, and then wait 20 to 30 minutes for your skin's pH to naturally neutralize before you apply Palmitoyl Tripeptide-38. Alternatively, you can just separate them by routine. Use your acids at night and your peptides in the morning. And remember, peptide results are transient. The newly synthesized collagen will last for months, but hyaluronic acid and laminin regressions will begin just weeks after you stop using it.
Section 5. Strategic synergies and stacking. Let's wrap up by looking at how to properly position this asset alongside other active ingredients in a routine. This competitive matrix clarifies exactly when and why you deploy Palmitoyl Tripeptide-38 over the others. Matrixyl pentapeptide 4 is highly targeted just for collagen 1 and 3. Argireline focuses specifically on dynamic muscle relaxation. GHK-Cu handles broad wound healing. But Palmitoyl Tripeptide-38 is your go-to choice for broad extracellular matrix coverage across 6 distinct components. And because it doesn't compete for the exact same receptors, it actually pairs exceptionally well with those other targeted assets.
Which leads us nicely to this optimized multi-layered protocol. You can build a truly comprehensive structural routine by stacking niacinamide at the top. This boosts the NAD energy levels required for protein synthesis. In the middle, Palmitoyl Tripeptide-38 acts as your core asset, rebuilding the dermal-epidermal junction and that structural scaffold. And down at the base, argireline pauses the mechanical folding of your expression muscles, which allows the dermis above it to rebuild without constant continuous structural stress.
To summarize our core takeaways from this evaluation, first, the breadth advantage. PT38 is the definitive general contractor of peptides, coordinating 6 separate ECM components. Second, the formulation imperative. It absolutely demands strict adherence to a 3 to 5 parts per million active concentration and a stable pH environment of 4.5 to 6.5. And third, the portfolio strategy. Its non-competing mechanism makes it the perfect foundational structural layer to stack alongside energy boosters like niacinamide and expression inhibitors like argireline.
So, I'll leave you with a final thought to evaluate your own formulations. Are you calculating the true parts per million of your peptide, or are you just trusting the marketing label? That's all for this episode. This is Peptide Proof. We'll continue tracking peptide science with rigorous, independent, evidence-based analysis, from research and synthesis to therapeutics, applications, industry, and market developments.